Hello, welcome to section 17.6 of chapter 17. This is on genetic mutations. I'm sure you've all heard of mutations, and you know the things you've heard of certain, let's say, hereditary diseases, genetic diseases, and such things. And these mutations occur in the in the genome, in the DNA. And of course, they could occur in, you could have errors in RNA synthesis as well, but that's going to be a one-off thing. It's not going to be a persistent thing that, it's going to just one protein molecule, which could be bad, but that's really not what we usually think of as mutations. We think of it in the DNA, in the genetic material. And one of the examples is they give here is the condition of albinism, which is when your skin or your body, your skin, your hair, does not produce melanin, and which is what gives our melanin and one or two other things, I think, give our skin its color. Well, when there's no melanin, the skin color, the hair color, and so forth is white because there's no pigment. And that's due to a mutation that prevents the body from, the skin, the body from producing melanin. So that is a genetic disease. We've already talked about another one. I think I'm pretty sure I mentioned, I know the text mentions sickle cell anemia earlier on, which is a mutation in the hemoglobin. And now we're going to talk about what mutations are chemically, let's say, and how they work in the different types. Mutation is a change in the nucleotide sequence of DNA. That's pretty much it. You already know about the genetic code. You know that proteins are synthesized by a messenger RNA being formed from a DNA template, and that goes onto a ribosome, the messenger RNA serves as a template for the formation of the synthesis of a protein, the translation process. And you change the nucleotide sequence of DNA, you might alter the sequence of amino acid sequence in a protein. And that could affect the structure and function of protein in a cell. Could make it better, could make it worse, could inactivate it, could do a lot of things. Mutations can come from a lot of different sources. X-rays, which an X-ray will hit a base in a nucleotide and change its structure, which means it's going to change how it pairs with the next time the DNA replicates, or when that gene is being read, it's going to be read as something else other than it should be. Overexposure to the sun, which is ultraviolet or UV and ultraviolet are the same thing. Very high energy radiation, so it can cause mutations. Chemicals called mutagens. There's a number of these things. There's things like there's a lot of carcinogens they can insert themselves between two nucleotides and mimic a nucleotide, so that it's almost like an insertion there. There's alkylating agents will put say a methyl group somewhere where it doesn't belong, and there's quite a few things that can be mutagens. Some viruses can cause mutations. In fact, give you a little story. In the human placenta, there's a protein which jumped over into humans primates, around 50 million years ago, somewhere around there. And it just so happens that protein is very, very useful in allowing the placenta to form and to do what it does. All right. And that made it possible for mammals to be, female mammals to carry their young until they're ready to be born, which has some advantages. You can move around. It's not like a bird sitting on a nest has to stay there until the birds hatch and are raised. You can actually move along. You take your nest with you, as you were, or take your young with you. It made it a little. It was an advantage for mammals. Okay, that is one of the things. And then there's other viruses can also cause mutate. Can actually just do other chemical things too to cause cancer. So, but insertion of genetic material is one thing that viruses will do occasionally. We're going to talk about that. If you have somatic cells and you have germ cells. Okay, somatic cells are body cells. Germ cells are the cells, eggs and sperms. All right. Their germ cells transmit whatever's in them, whatever genetic information is in them, to the offspring. Somatic cells don't do that. A mutation in a somatic cell is only going to affect that particular organism. If it causes if mutations in a germ cell, then all the DNA from in a new individual, or well, if it's in one germ cell, the DNA produced in that new individual is going to be affected by the genetic change. And mutations that cause uncontrolled growth can cause cancer. All right. But it's not the only, cancer could be genetic. It could be due to mutations. A lot of other things can be due to mutations as well. At the end of this section, we're going to see a table of a number of genetic diseases. Now, there are several types. You're already familiar with. I'm going to go to the next slide just to make a point here. You notice variants of this slide are going to be used in the next several slides, and this represents a normal DNA and the protein synthesis from it. Okay, we know from the last section how that works. You have a DNA template forms a messenger RNA in the process of transcription, which is translated to amino acid sequence. All right, so that nucleotide sequence, the T's and A's and U's and G's and so forth, that is going to be ideally fixed in the genetic material. All right, so we'll come back to that. A couple of things can happen here. You can have what's called point mutations, which is the replacement of one base by another in the DNA. That can do a couple of things. It can do nothing. And remember, if you go look at the genetic code, let's say you have, I'm looking at this table 17.5 right now in your text, and we've said that leucine, there's six codons for leucine, and let's say we have the CUU, the cytosine uracil codon, okay? And if there's a point mutation there, if the second U it mutates to let's say a guanine, you have a CUG, and so you have a mutation there. You've changed your genetic material, but that codon still codes for leucine. So it's called a silent mutation because you don't see an effect. It doesn't produce a change in the amino acid sequence. Now, we'll take the CUU, and let's change instead of the second U, let's change the first U to a G. That means you're going to have a CGU, and now that's going to be arginine, CGU, and that's going to be a big change. Yeah, arginine's a basic amino acid, and leucine's a hydrophobic amino acid. So that's going to be a significant change. So a mutation can be silent, or it can actually change the amino acid that's going to be put into the protein chain when that gene is expressed. You can have a deletion mutation, in which a base is deleted from the normal order of bases. In other words, there's one base is removed, so one's missing. So the reading frame is going to be shifted in this case, and we're going to see that when we see these next few slides. An insertion mutation does the same thing. It shifts the frame, but now there's another base goes into the DNA and shifts it that way. So we're going to look at these now in the next few slides. Okay, so we see the normal one here, and right around here is where they're going to be playing around here. So just this is AAACC. They're going to be mostly working there, giving you mutations. Okay. So I have a point mutation. Now say this was a C, a cytosine, and now it's been replaced by thymine in the DNA. Remember, thymine in the DNA, uracil in messenger RNA. And when a messenger RNA is made, you see that where we had a guanine in the guanidine phosphate nucleotide here. Now you have adenine. Now you have adenosine here. Adenine is the base. Adenosine phosphate is the nucleotide. And you see that's going to change the amino acid, which was glycine, which is GGC, to AGC, which is serine. So that's a substantial change. You've gone from essentially a nonpolar amino acid to a very polar one. So that will have some effect on the chemistry of this. Things like leucine, isoleucine, or to valine, between valine and alanine or something like that. They will have an effect, but oftentimes it will still be a functional protein. So we see these deletion mutations, insertion mutations. This time now, the insertion mutation, you see there were these three adenines here. Now you're putting a thymine, inserting it in there, and now you're changing. Okay, there's a deletion mutation. Now you've shortened the chain, so you've shifted this over with the deletion. Now you've got leucine and alanine where you had phenylalanine, serine, and a stop codon. So you may not have a stop codon anymore here. It looks like you don't. UAA. Okay, yeah, see this U here, it was UAA here, now this is no longer a stop codon because this U is now has been pulled back to an earlier place in the line, and so it's in a part of an alanine codon, and now the AA that had been part of a stop codon are now part of something. A and the two A's were part of a stop codon, they're now something else. And in this case with the insertion mutation, you see you've changed. You've gone from phenylalanine serine stop to leucine, tryptophan, leucine. Now you're making big changes here in the structure of the protein. And that, some organisms can get away with that. And sometimes actually, if you look at, there's compilations, there's tables of various proteins that are what they call homologous proteins, where you have essentially the same protein in different organisms. And hemoglobin's a common one. You'll see tables, you'll see a table of hemoglobin where, you know, the sequence of human hemoglobin, chimpanzee hemoglobin, and human and chimpanzee hemoglobin are identical. But, you know, you have gorillas, you know, you have the apes, you know, the primates, they're all really close. And then you get further away, like horses and chickens and stuff like that. And you see more and more differences in the amino acid sequence. And sometimes you get insertions, and sometimes you have whole blocks of amino acids inserted. So there's a lot of variations on this. We're mostly only worried here about just a single mutation. You're going to change when you change an amino acid sequence, a silent mutation, of course, isn't going to really change anything. If the same codon mutates again, it can either, you know, stay the same, or it can come up, you know, it can give you, maybe, you know, the first mutation might make the next mutation more prone to make a change or something like that. Maybe not, but again, we're not going that far. When the mutation changes the amino acid sequence, as we've said and seen, the structure of the resulting protein can be very different afterwards, and it could cause a loss of its biological activity or some alteration of its biological activity. And when this condition is hereditary, that means when the condition is inherited in the germ cells, it was in the germ cells of the parents, this is going to be caused, and of course, if it was in the germ cells of the parents, it's going to be in the germ cells of the offspring, and it's going to be transmitted. This is a genetic disease. So here you have the sequence where you have DNA, and you have some kind of mutagen, you know, X-ray, sunlight, chemical mutagens, viruses, alters the DNA, gives you a defective protein, a defective gene. If that defect gets into the germ cell and it's transmitted to the offspring, you can have, first of all, you can have a genetic disease. And if it's not in the germ cell, if it just is in a somatic cell, it can turn into cancer. There's quite a few genetic diseases, and they give a couple of examples here, and then there's a table that I'm going to go through pretty quick. But it's usually going to be something like a defective enzyme. All right, so you have what's called phenylketonuria, which it's a reasonably common one. The DNA cannot direct the synthesis of the enzyme phenylalanine hydroxylase. In other words, there's something wrong with the gene for phenylalanine hydroxylase, and either the enzyme is not made, or the enzyme's defective and doesn't do its job, which is converting phenylalanine to tyrosine. And that means what happens is that there's going to be a buildup of phenylalanine in the usually this is going to be spotted in childhood, and there's ways of treating it. You need to keep a low phenylalanine diet, but it can be managed, but it's still serious. And then there's albinism. We saw that, where they mentioned the synthesis of melanin. Well, melanin is synthesized from tyrosine. So when that enzyme is defective, then you're going to find the organism is going to be an albino. Okay, we're actually done with this section. And we're finishing up with a list of common genetic diseases. You've probably heard of some of them, maybe not of others. So you have galactosemia, where there's a defect in the metabolism of galactose 1-phosphate. We didn't really see galactose too much, except in the context of it being a component of lactose. bonded with, it was lactose, as you recall. It was a disaccharide with galactose and glucose. Cystic fibrosis, that's another one that you've probably heard of this one, but there's a protein that regulates production of stomach fluids and mucus, and of course thick mucus secretions make breathing difficult, and they also interfere with the pancreas. So that's one of the more common inherited diseases in children. Again, we've probably heard of Down syndrome, which is a result, your chromosomes usually have pairs of chromosomes. Well, Down syndrome, it's usually number 21, as they say here. You'll have 3 chromosomes instead of just 2, so you're going to have a triplet of chromosomes, and that causes various levels of defects: heart, eye, mental, physical. Then there's familial hypercholesterolemia, where you make abnormally high cholesterol levels, which is bad. Muscular dystrophy, Duchenne's muscular dystrophy, which is this is in one of the in the X chromosome, so it's transmitted by the mother, and or actually no, men are XY, so they can transmit either an X or a Y. This is extremely painful, and usually the sufferer dies by age 20, about 1 in 10,000 males, which doesn't sound like a lot, but. And there's Huntington's disease, which I believe Huntington's is a dominant gene, the gene for that, which means that if it's, there's dominant and recessive genes. Recessive genes, if you have a pair where there's you get one gene from the mother, one gene from the father, and you've got the dominant gene is going to be the one that's going to be expressed in a case like that. And if you have the recessive gene from both parents, then you definitely will have that condition. I don't know that for sure. I'm pretty sure it's Huntington's, but it's either Huntington's or Duchenne's. And then you have sickle cell anemia. We talked about that when we were talking about proteins. Hemophilia, where you have a blood clotting factor is defective, so blood doesn't clot properly. And the one that I wanted to mention here was Tay-Sachs. That is the result of a defective hexosaminidase A, which causes an accumulation of gangliosides. We didn't talk about gangliosides when we were doing lipids, but a ganglioside is similar to a sphingomyelin. The difference is that you might recall the sphingomyelin has a phosphate linked with an amino alcohol, and rather than the phosphate and amino alcohol, you have on that, there's a hydroxyl on the sphingonine, or sphingosine if you prefer, and there's a hydroxyl that is linked with a carbohydrate, and in the hexosamine it's a N-acetyl uramic acid, something like that. But we're not, as I said, we're not worried too much about that, but I figured I had to tell you what a ganglioside was. Okay, let's go through a learning check here, where we identify each type of mutation as a point, deletion, or insertion. Okay, cytosine C enters the DNA sequence. Well, it enters the sequence. That's going to be an insertion mutation. One adenosine is removed from the DNA sequence. Going to be a deletion. the base sequence goes from TGA to TAA. That is a point mutation. As you see, and there we have it. Okay, we're done with section 17.6. I'm going to move on to 17.7, and we're going to try to finish this chapter and have this complete set for you.